See component 1 task book, and we're in the respiration section, and we are on question 2. The diagram of ATP. Right, we've seen hundreds of these before. Done some past paper questions, you'll have seen many of these. Label the parts of the structure. That's a gift for two marks, isn't it? It says name. Okay? If we said name you, we wouldn't say human, we'd say your name. So it actually wants the name rather than the type, although they're probably being a little bit generous. So this actually, the name of it is adenine. Although we could say it's a nitrogenous base and so on, but it wanted us to name it. And likewise, it is ribose. Although it will accept pentose or a 5-carbon sugar. Describe the use of ATP in the process of respiration. Lots of people have read this wrong and not understood this and thrown away these 2 marks. Describe the use in respiration. So when is it actually used in respiration? Well, let's name the stage. So it's actually used in glycolysis. So you might remember glycolysis produces 2 ATP overall. It actually produces 4, but 2 get used, so 4 minus 2 is 2. So it's actually used in glycolysis, and it phosphorylates our glucose, or sticks phosphates onto glucose, and that activates it and makes glucose more likely to react in the next steps. So phosphorylation of the substrate. If we put glucose on, I might as well mention it. We've said our 2 marks now, but there's an activation of the substrate. So it's amazing how many people just give us a use of ATP after respiration and then throwing away those 2 marks. And describe the production of ATP by chemiosmosis. Well, you either know this or you don't. It's just straight out of your notes. And we've already just really done it in the previous question. So it's just the same sort of thing. Let's talk about our proton gradient. So H plus gradient, from the intermembrane space to the mitochondrial matrix. Again, say how we've got to that gradient. So powered by the electrons, the electron transport chain, the electron transport chain. What happens to these? They diffuse, protons diffuse through that stalked particle. Contains ATP synthetase containing ATP synthetase and production of ATP. So we get ADP plus our inorganic phosphate going to ATP. So that is an absolute gift. If you see a question like that, 4 marks, nearly half an essay. It'll take you just a couple of minutes to reach into your brain, retrieve the section of notes and stick it down. No problem. Over the page, it gives you the structure of NAD, which you might not know. and it says a molecule of NAD. It looks a little bit like it's 2 ATPs, or well, it won't be T, but there's only one of them joined together. But other than that, it looks like nucleotide and another nucleotide. So it's a dinucleotide. And how it differs. So how is this one different? Well, this one has got two phosphates versus 3 in ATP. So I'm actually talking about this molecule rather than ATP. This has got 2 sugars, whereas ATP has only got 1. So this has got 2 sugars, or 2 riboses, versus 1 in ATP. This has got 2 bases. So once you've started along this, it's quite easy, isn't it? 2 bases versus 1 in ATP. And you could say it's two nucleotides versus 1. That sort of thing. So hopefully pretty straightforward that. Describe the use of NAD in aerobic respiration. A little bit more tricky this. And if you've done the previous question, that might give you a little bit of inspiration. What's going on there is carrying hydrogens onto this next stage, and that's essentially what we need to describe. So it accepts hydrogen. Or another word for that, it becomes reduced. So it becomes reduced, if you like chemistry. We all like chemistry, don't we? And what else can we mention here? So say if we're struggling for what's going on, well, how have we got our hydrogen? Let's mention our enzymes and where things happen and things like that. Becomes reduced. We can mention dehydrogenase enzymes used. So we're just trying to scrounge these marks really. Where does this happen? Well, this happens where we're taking this hydrogen. So passes the hydrogen and the electrons, because remember hydrogen is a nucleus that contains a proton and an electron. So our proton is H+, so it's H+ and an E-, and it's passing these on and then being recycled itself. So let's talk about our hydrogen. Passes H+ to the inner mitochondrial membrane, where it'll get pumped, and we could say passes. Let's talk about our electrons. Electrons, E-, let's put electrons to the electron transfer chain, ETC, for ATP synthesis. That's the job it's doing. And then of course the last bit it's actually doing is being recycled again. So where's that little diagram? You can see there it's being recycled back to NAD, so it can pick up more hydrogens. And then you could say reoxidized or recycled to accept more further hydrogen. So a little bit tricky in the end, coming up with stuff. But if you're not sure, you get the first one, it becomes reduced or accepts hydrogen, but then you just need to say things that you know around that and try and scrounge some marks.